Hi class, this is your instructor, Skylar Huff, and we're now in Chapter 16, Human Genetics. So this also includes the human genome, as we previously learned about in the last chapter. The principles of genetics apply to all organisms, including humans. However, important differences separate genetic research on humans and genetic research on other organisms. To study inheritance in other species, geneticists may conduct controlled mating between individuals and raise their offspring under carefully controlled conditions. Of course, setting up experimental matings in humans is unethical and illegal. Despite the inherent difficulties of studying inheritance in humans, human genetics, the science of inherited variation in humans, is progressing rapidly. And just one example of the rapid progression involves what you all see to begin the chapter on page 340. So as you look there on page 240, it's about, of course, looking at the rapid chromosome mapping. So this is the fluorescence of a human chromosome. So it has 10 different colored fluorescent probes attached to specific DNA sequences. And this technique allows researchers to efficiently determine certain chromosome rearrangements and abnormalities. So the medical attention given to human genetic diseases has expanded our knowledge of human genetics. Our most detailed understanding comes from diseases caused by defects in single genes that exhibit a Mendelian inheritance pattern. Valuable insights have come from studies of other organisms that have similar genes, and many human inheritance questions have been explained using model organisms such as bacteria, yeasts, worms, fruit flies, and mice. Researchers also conduct population studies of large extended families with a high incidence of a particular type of human genetic disease. These studies use genetic information and technologies derived from the Human Genome Project to uncover genes associated with many different types of complex genetic diseases. For these diseases, many genes interact to cause various conditions, including susceptibility to certain types of cancer, diabetes, kidney disease, obesity, and behavioral disorders such as schizophrenia, depression, and autism. Here, in this chapter, we'll first examine methods of human genetics, including new research directions based on DNA technologies discussed in our prior chapter, being chapter 15. And we'll then discuss a variety of human genetic disorders, and we'll explore the use of gene therapy as well as the application of genetic testing, screening, and counseling for families at risk. And we'll even get to a conclusion with the ethical issues related to human genetics. So let's begin it formally, class, here with studying human genetics. And with this, just keep in mind that human geneticists use a variety of methods that enable them to identify genetic defects and to make inferences about a trait's mode of inheritance. So we'll consider three of these such methods: the identification of chromosomes by karyotyping, the analysis of family inheritance patterns using pedigrees, and DNA sequences, and mapping of genes through genome projects. So investigators often study human inheritance most effectively by combining these and other approaches. So human chromosomes are studied by karyotyping. And I'm quite confident, and this is the figure I mentioned earlier to start the chapter from page 340. Back to where I was meaning. Or back to where I had begun, excuse me. So some well-known human genetic disorders involve changes in the structure or number of chromosomes, and the normal number of chromosomes for the human species is 46. It's made up of 44 autosomes, or 22 pairs of autosomes, and 2 sex chromosomes, or a pair of sex chromosomes. So a karyotype illustrates an individual's chromosome composition, showing both the chromosome number and any large structural defects in the chromosome. So human chromosomes are visible only in dividing cells, which you all learned about in the 10th chapter with the process of mitosis by way of the cell cycle. And what I'm getting at is they are difficult to obtain directly from the body. So researchers typically use blood cells because white blood cells can be induced to divide in culture medium by treating them with certain chemicals. So other sources of dividing cells, including skin and for prenatal chromosome studies by way of chorionic villi or fetal cells shed in the amniotic fluid. So in karyotyping, a karyotyping biologist culture dividing human cells and then treat them with a drug called colchicine, which arrests the cells in a mitotic metaphase or late prophase when the chromosomes are most highly condensed. Next, what researchers then do is immerse the cells in a hypotonic solution. So the cells will then swell and the chromosomes will spread out and are easily observed. And you can think back to what you all learned about cellular transport and the process of osmosis, and especially how cells behave by way of tonicity in those three different types of solutions. So the investigators then flatten the cells on microscope slides and stain the chromosomes to reveal patterns of bands, which are unique for each homologous pair. After the microscopic image has been scanned into a computer, the homologous pairs are then electronically matched and placed together. So by convention, geneticists identify chromosomes by length and, of course, the position of centromere, the banding patterns, which are produced by aligning chromosomes with dyes that produce dark and light cross bands of varying widths, and other features such as satellites, which are the tiny knobs of the chromosome material at the tips of certain chromosomes. So with the exception of the sex chromosomes, all chromosomes, but chromosome 21, which is smaller than chromosome 22, are numbered and lined up in order of size. The largest human chromosome is chromosome 1, and it is about 5 times as long as the smallest chromosome, which of course is chromosome 21. But there are only slight differences among some of the intermediate sized chromosomes. The X and Y chromosomes of a normal male are homologous only at their tips. Normal females have 2 X chromosomes and no Y chromosome. So differences from normal karyotype, that is, and of course, deviations in chromosome number or structure are associated with certain disorders such as Down syndrome, which of course we will get to, class, just a bit later. So what you all have here for you is what a normal karyotype looks like. And yes, this is for a male. And if you're saying, well, how do we know it's from a male? Excuse me. Well, the way in which it's known that's from a male is by looking for that Y chromosome right there. So the presence of that chromosome is all you need to know whether or not this is from a male or female. So with this being the case, I'll next get to another way to distinguish chromosomes in a karyotype, and this is by fluorescent in situ hybridization, called FISH. So a geneticist tags a DNA strand complementary to DNA in a specific chromosome with a fluorescent dye. So as that happens, the DNA in the chromosome is denatured. That is, the two strands are separated so the dye strand can bind to it. A different dye is used for each chromosome, painting parts of it with each color, which of course is what you all are seeing here. And this technique can, of course, be used to detect breakage and fusion of chromosomes and abnormalities associated with certain genetic diseases and many types of cancers. So let's get on into what's next, and that reminds me. So on your second of 2 lab tests, just keep in mind that you will be expected to, of course, look at a karyotype and make determinations of course, whether or not the karyotype is normal or even from a male or a female. So by the time this chapter has ended, you will know exactly, class, where to look and what to look forward to in order to perform well in that second of 2 lab tests. So the family pedigrees, they help identify certain inherited conditions. Early studies of human genetics usually dealt with readily identified pairs of contrasting traits and their distribution among members of a family. So the family tree shows the inheritance patterns, meaning the transmission of genetic traits within a family over several generations is a pedigree. So the pedigree analysis, it remains widely used, even in today's world of powerful molecular genetic techniques. And it's because it helps the molecular geneticists determine the exact interrelationships of the DNA molecules they analyze from the related individuals. So pedigree analysis is also an important tool of genetic counselors and clinicians. So, however, because human families tend to be small and information about certain family members, particularly particularly deceased relatives may not be available. Pedigree analysis does indeed have its limitation. And I'm sorry, I meant particularly in deceased relatives, not diseased relatives. So pedigrees are produced using standardized symbols. So with that being the case, let's get to at least an example showing some of those standardized symbols. So this is figure 16.2 from page 342. So it shows, class, the pedigree for albinism, which of course is because of a lack of the pigment melanin in the skin, hair, and eyes. So each horizontal row represents a separate generation, with the earliest generation being at Roman numeral 1. And I have to change my color. So this is that first generation at Roman numeral 1. Of course, the second generation there at Roman numeral 2. And the third and final generation, at least here, class, at Roman numeral 3. So, as I was stating, class, the earliest generation at the top and the most recent generation at the bottom. So within a given generation, the individuals are usually numbered consecutively from left to right using Arabic numbers. So, of course, that's what you're seeing here. So from left to right, from 1 to 4, and of course from 1 to 5, and here, class, from 1 to 4. A horizontal line that connects two parents and a vertical line drops from the parents to their children. For example, individuals 2, 3 and 2, 4 are parents of 4 offspring, being 3, 1, 3, 2, 3, 3, 3, 4. So I was saying individuals 2, 3 and 2, 4 are parents of 4 offspring, being 3, 1, 3, 2, 3, 3, and 3, 4. And of course, within a group of siblings, the oldest is on the left and the youngest is on the right. So let's get into the biology here. The allele for albinism cannot be dominant. If it were, let me put that comma there, at least one of the female 2's parents would have been albino. For instance, I was saying that if in fact, class, this here was inheritant, and it was inheritant, class, as a dominant disorder, at least one of the alleles for her parents would have to, of course, had that allele, and of course, they would have been albino. That's not the case. So that's how you know, and that's how you can see, class, that the inheritance of albinism is not and cannot be dominant. So, of course, this pedigree is explained if albino is inherited as an autosomal, meaning not carried on the sex chromosome, recessive allele. So in such cases, 2 phenotypically normal parents could produce an albino offspring because they are and as I said, they are heterozygotes. I'm sorry about that announcement there. So as it says, they are heterozygotes, and of course could each transmit a recessive allele. So studying pedigrees enables geneticists to predict how phenotypic traits that are governed by this genotype at a single locus are inherited. So more than 1,200 traits have been described in humans. Pedigree analysis most often identifies 3 modes of single locus inheritance. In other words, it could be autosomal dominant, autosomal recessive, and X-linked recessive. So we'll get to these examples and modes of inheritance a bit later in the chapter. So let's continue on to, of course, the gene database. So human gene databases allow geneticists to map the locations of genes on chromosomes. So as this was done, meaning although the pedigree analysis can provide information about the type of inheritance associated with a single gene, it does not provide the specific information about where that gene is located or how that form of the gene causes the disease. Furthermore, most genetic diseases involve many genes that are produced in complex patterns of inheritance. So how do you identify a specific gene and find its location in an extremely complex genome if you know nothing about it? Well, using the database of the Human Genome Project and other studies, researchers have mapped and generated, of course, those maps of each chromosome that shows the precise location of each gene. And class, this is what's pretty amazing here. It's known, of course, the location of those genes on those chromosomes, period. So these maps allow investigators to locate the region where the gene resides on a specific chromosome and then to analyze the genes within that region to determine which one is associated with the disease. Once the disease-causing gene is identified, geneticists can initiate studies to determine its function and how it interacts with other genes associated with the disease. So single nucleotide polymorphisms are used as genomic landmarks to determine and locate genes linked to genetic diseases. So these single nucleotide morphisms are single letter differences in DNA sequences, and they're found every few hundred bases within the genomes of individuals in large populations as they are compared. So in 2015, the 1000 Genomes Project, which of course is made up of a group of scientists in 8 countries, published data derived from 1,092 individuals from 14 different populations, providing a map of 84.7 million single nucleotide polymorphisms and 3.6 million short insertions and deletions that span all human chromosomes. So investigators used DNA chips and whole genome sequences methods to compare the distribution of more than 1 million single nucleotide polymorphisms in DNA isolated from individuals by a given genetic disease with those who are not affected. So by identifying a specific single nucleotide polymorphism that consistently appears in the affected individuals, but that is rare in unaffected persons, the researchers can locate a small region of the genome that contains the gene now associated with that disease. So there it is. So genome-wide association studies, which of course gets us back to, well, this shows class, the normal, and of course that from the diseased individual, or at least an affected individual, excuse me. That's that difference. It's just a single difference. And of course, you can then see that single nucleotide polymorphism. The next we go. So GWAS. So in humans, only about 1.5% of the genome, meaning class, that's very small, meaning of all of those billions of base pairs, it's only about 1.5% of such that specifies amino acid sequences of proteins or polypeptides. That should be mind-blowing. Of course it is to me. That, of course, it's only a percentage class. It specifies proteins. However, reports from the ENCODE project have shown that at least 80% of the human genome is involved in biological functions. So these DNA sequences include non-protein coding regulatory elements and non-protein coding genes that produce RNAs with unknown functions. So many times RNA coding genes are found in introns as well as in the regions between genes that were previously thought to be gene deserts or junk DNA with no biological function. Look at that. So researchers are now actively engaged in genome-wide association studies, or GWAS studies, to compare the genomes of individuals with a particular disease to individuals without that disease. And in one study, for example, the researchers used single nucleotide polymorphism analysis to survey the genomes of more than 14,000 British people to compare the genetic patterns in healthy people with those in people with one of 7 common diseases. The phenotypes of these diseases exhibit complex inheritance patterns with many different genes contributing to the susceptibility. In the study class, they identified 24 single nucleotide polymorphism variations associated with bipolar disorder, one with coronary artery disease, 9 with Crohn's disease, 3 with rheumatoid arthritis, and 7 with type 1 diabetes, and 3 with type 2 diabetes. So that's a lot of information, class. That was, of course, gleaned from such thing. So with this, just keep in mind that many genetic variations with complex human diseases are found in non-protein coding genes. And the way in which it's happening, that a researcher class can determine the function of disease-associated genes is, well, it's done, class, meaning just the first step toward developing treatment for the disease. And then once they develop the effective treatment strategies, the scientists must first identify that specific gene that causes the disease. And of course, they must have that function. So comparative genomics has revealed that DNA in both the mouse and human genomes. And with that comparison, the genomes of both of which, they are about 500 DNA segments longer than 200 base pairs and are 100% conserved. I'll say it again. A comparison between the mouse and human genomes has shown that about 500 DNA segments longer than 200 base pairs are 100% identical, class. This is a remarkable amount of conservation. And of course, it has interesting evolutionary implications, which mean, of course, that these segments have not mutated in the past 75 million years since the mouse and humans shared a common ancestor. And it is during this time that the other DNA sections underwent considerable mutation and selection, allowing mice and humans to diverge to their present states. So although the functions of these unchanged elements are not yet determined, they clearly have a vital role. And if they are not essential in their present form, they would have already undergone mutation and selection forces. So these strong evolutionary connections allow investigators to use the mouse as a powerful research tool to detect and investigate the functions of human disease-causing genes that are common between the two species. Hence, class, the mouse is always going to be, of course, used. It's always used as that model. So researchers use the mouse models to study human genetic diseases, as I just stated. So many questions relating to human genetic diseases are difficult to answer because of the ethical issues involved in humans as test subjects. However, on any disease, it's greatly facilitated if an animal model is used for experimentation. So a good example, class, is with cystic fibrosis, a genetic disease caused by a single gene mutation inherited as a recessive allele. Researchers use gene targeting to reduce strains of mice that were either homozygous or heterozygous for cystic fibrosis by replacing the normal mouse genes with ones containing the same types of mutations found in human cystic fibrosis patients. The allele that causes cystic fibrosis is a mutant form of a locus involved in controlling the body's water and electrolyte balance. Geneticists have cloned this gene and found that it encodes for a protein. It's called the CFTR protein, and it serves as a chloride ion channel in the plasma membrane. And if you're wondering what CFTR stands for, it stands for cystic fibrosis transmembrane conductance regulator. So this ion channel provides chloride ions out of the cell's lining. And I'll say it again, this ion channel transports chloride ions out of the cell's lining of the digestive tract and the respiratory system. So when the chloride ions leave the cells, water follows by osmosis. Thus, the normal secretions of these cells are relatively watery because the cells of individuals with cystic fibrosis lack normal chloride ion channels. The individual's secretions have a lower water content and their sweat is very salty. Cells of heterozygous individuals have only half the usual number of CFTR ion channels, but they are not enough to maintain normal fluidity of their secretions. So using these mouse models, researchers have focused on understanding the way in which the CFTR channel is activated or inactivated. So with this information is used to design drugs that enhance chloride transport through the CFTR channels, and drugs have been recently developed to be able to treat some types of cystic fibrosis in humans by activating the mutant channels. So this is pretty interesting. So let's get on into chromosomal abnormalities class. So chromosomal abnormalities can, of course, well, be abnormal in both number and structure. Polyploidy class is the presence of multiple sets of chromosomes, and this, of course, is common in plants, but is so rare in animals. So it may arise from the failure of chromosomes to separate during meiosis, or from the fertilization of an egg by more than one sperm. So when it occurs in all the cells of the body, when it occurs in all the cells of the body, polyploidy is lethal in humans and many other animals. For example, triploidy or triploidy is sometimes found in human embryos that have been spontaneously aborted in early pregnancy. And I'll be blunt, class, and this is good to know, but many times, class, pregnancies are spontaneously aborted, and it's not even known that the person had been pregnant at all. And this, is, of course, has been found, meaning triploidy, 3N, because, of course, it should be it should be diploid, period. Nothing else, of course, in human embryos. So next up, the abnormalities caused by the presence of a single extra chromosome or the absence of a chromosome is called aneuploidy. And if you want to write that down, class, being off by one chromosome, well, that is definitely what aneuploidy is. So a more common and aneuploidy class, of course, they are more common than polyploidy. So disomy, of course, is what's normal. Disomy being you have two of each kind of chromosome in a cell or individual, whereas in trisomy, a cell or individual has two copies of each chromosome except for one, such as you all may have known about, class, with trisomy 21, or I guess you say trisomy 18 or 16. And just to make sure I go back to aneuploidy one more time, just keep in mind, class, it's off by one chromosome, so it could be either A, 2N plus 1, which of course I just mentioned as being trisomy, or 2N minus 1, causing a monosomy, which is where I'm going in just a moment. So with that being the case, the trisomic chromosome number is designated as 2N plus 1. In monosomy, an individual lacks one member of a pair of chromosomes, so a monosomic chromosome number is designated, class, as 2N minus 1. And if you look closely, class, at table 16.1, it goes through a number of disorders that aneuploidies produce, such as I mentioned, trisomy 18 or even trisomy 21. So trisomy 18 would, of course, cause someone to have Edwards syndrome, and trisomy 21 would cause someone to have Down syndrome. Trisomy 13 would cause someone to have Patau syndrome. XO will be Turner syndrome. That's that person having only that X chromosome, not XY, not XX, not XXY, and not XXY, and not XXX, but only class X. Klinefelter syndrome, class, is XXY. And then you have XYY. And then finally, class XXX, which is triple X. All right. So getting back to how aneuploidies, class, are produced, or at least how they have come about. So aneuploidies generally arise because of an abnormal meiotic division. And as that does occur, the chromosomes fail to separate at anaphase. So failure to separate, class, of course, will end up with far too much, I would say, genetic information. So the phenomenon is called nondisjunction, and it can occur when the autosomes or the sex chromosomes in meiosis, or the sex chromosomes, excuse me. So in meiosis, chromosome nondisjunction may occur during the first or second meiotic division, or even both. For example, two X chromosomes that fail to separate at either the first or second meiotic division may both enter the egg nucleus. Alternatively, the two joined X chromosomes may go into a polar body, leaving the egg with no X chromosome at all. So next up, this shows it. So if you look closely with nondisjunction in that first meiotic division, so we have X here, we have Y there, nondisjunction occurs. Due to nondisjunction, that gamete has X and Y, that gamete has X and Y, and these class here have nothing. And of course, the same is shown there. So it's very important class to know about how nondisjunction occurs. And this class shows it yet again. So the first meiotic division, so we have X there, we have Y there, both of course in that same cell. So with our first division, it occurs as it should, normally. Here, class, in that second division, before it occurs, of course, there it is. The chromosome has failed to separate. Separation occurred here. So, of course, these are normal. Normal. However, this has nothing. And there it is, all because of nondisjunction. That class would then cause there to be far too much genetic information. And of course, if it were to be in the Y, in the second division class, it would look like this here. So Y is there, nothing is here, and of course, still X. So the showing you class just ways in which nondisjunction could occur. So nondisjunction of the XY paradigm in the first meiotic division in the male may lead to the formation of a sperm with both X and Y chromosomes, or a sperm with neither NX or Y, as we saw class moments ago. X and Y in the same sperm cell, or a sperm cell class with nothing. Similarly, class, nondisjunction in the second meiotic division can produce sperm cells with two X's or two Y's, which of course we see here, two X's class and NB. So an abnormal gamete unites with a normal one. The resulting zygote has a chromosome abnormality that will be present in every cell of the body. So meiotic nondisjunction results in an abnormal chromosome number at the zygote stage of development. So all cells of the individual will have that abnormal number of chromosomes. In contrast, nondisjunction during a meiotic division occurs later in development and leads to the establishment of a clone or abnormal cells in an otherwise normal individual, such as the mixture of cells with different chromosome numbers may or may not affect those body cells, those somatic cells, or germline cells, being those reproductive cells. So with this, we're now going to get to effects of these abnormalities. So different chromosome numbers are common in many cancer cells, particularly those in solid tumors. Whether aneuploidy is a cause or a consequence of cancer, however, is not yet clear. So the recognizable chromosome abnormalities are seen in less than 1% of all live births. I say again, less than 1% of all live births, you all. But substantial evidence suggests that the rate at conception is much higher. So at least 17% of recognized pregnancies will end in spontaneous abortion, as I earlier mentioned, class, meaning a miscarriage. So approximately half of these spontaneously aborted embryos have major chromosomal abnormalities, class, including autosomal trisomies, such as trisomy 21, triploidy, tetraploidy, Turner syndrome, in which, of course, that XO refers to the absence of a second chromosome, as I earlier explained. And then you, of course, get to autosomal monosomies, which are exceedingly rare, possibly because they induce spontaneous abortion, class. And this is very early in the pregnancy, before a woman is even aware that she is pregnant, as I mentioned, class, prior to now. So some investigators give surprisingly high estimates, being 50% or more. I repeat, 50% or more of the loss rate of very early embryos. That's good to know, class, and I say you should remember that. So chromosome abnormalities probably induce many of these spontaneous abortions or miscarriages. On to Down syndrome. So Down syndrome is one of the most common chromosome abnormalities in humans. And when I say syndrome, or when you see the word syndrome class, syndrome is what's referring to a set of symptoms that usually occur together in a particular disorder. So a syndrome is a set of particular symptoms that usually occur together in a disorder. So Down syndrome was named after John Langdon Down, who was a British physician who first described the condition. So affected individuals have abnormalities of the face, eyelids, tongue, hands, and other parts of the body and are intellectually disabled. They are also unusually susceptible to certain diseases such as leukemia and Alzheimer's disease. Studies of genetics class reveal that most people with Down syndrome have 47 chromosomes because of an autosomal trisomy. The condition class is known as trisomy 21. Nondisjunction during meiosis is responsible for the presence of the extra chromosome. And although no genetic information is missing, in these individuals, the extra copies of chromosome 21 genes bring about some genetic imbalance that causes abnormal physical and mental development. So Down syndrome is quite variable in expression, with some individuals far more severely affected than others. So researchers using DNA technologies have pinpointed genes on chromosome 21 that affect mental development as well as possible cancer-causing genes or oncogenes, and genes that may be involved in Alzheimer's disease. So like many human conditions, cancer and Alzheimer's disease have both the genetic as well as environmental components. So one cause of Down syndrome appears to be the result of the overexpression of chromosome 21 gene that encodes for microRNA. And I won't get so much into the microRNA portion, but what I get is the resulting underexpression of this gene class in a mouse model causes severe defects in neuron functions. So the incidence of Down syndrome class is 1 out of every 700 live births in the United States, and its incidence increases markedly class with increasing maternal age. So the occurrence of Down syndrome is not affected by the father's age, although other disorders are, including schizophrenia and achondroplasia, most commonly known as a form of dwarfism. All right. So Down syndrome is 68 times as likely in offspring of mothers age 45 than in the offspring of mothers at age 20. However, most babies with Down syndrome in the United States are born to mothers younger than 35. And this is in part because these women greatly outnumber older mothers. So the relationship between the incidence of Down syndrome and maternal age has been studied for decades, although there is no explanation. So scientists have proposed that several hypotheses to explain the maternal age effect, but none is supported unequivocally. One explanation is that older women have held eggs in suspended meiosis too long, leading to deterioration in mitotic spindle apparatus. And a woman is born with all of the oocytes she will ever have, and these oocytes remain in prophase 1 of meiosis until ovulation. So another possibility is that the aging uterus is less likely to reject an abnormal fetus than a younger uterus. So there you have it, and of course, here class, in figure 16.5a and b, you're seeing class phenotypically, as well as that karyotype with, of course, those 3 chromosomes there. Trisomy 21. This is Down syndrome. So on to sex chromosome aneuploidies. So most sex chromosome aneuploidies are less severe than autosomal aneuploidies. Sex chromosome aneuploidies are tolerated relatively well, at least in part because of the mechanism of dosage compensation. With dosage compensation, mammalian cells compensate for extra X chromosome material by rendering all but one X chromosome inactive. So the inactive X chromosome is seen as a bar body. So that's a region of darkly stained condensed chromatin next to the nuclear envelope of the interface nucleus. So next up is Klinefelter syndrome. And individuals with this, they are males with 47 chromosomes, including 2 X's and 1 Y. They have small testes, produce few or no sperm, and are therefore sterile. The hypothesis is that the Y chromosome is a major determinant in the male phenotype and has been substantiated by the identification of at least one gene on the Y chromosome that acts as a genetic switch directing male development. So males with Klinefelter syndrome tend to be unusually tall and have female-like breast development. So about half show intellectual disability, but many live relatively normal lives. However, each of their cells has one bar body, and on the basis of such, they would be erroneously classified as females. So about the incidence class is about 1 in 1,000 live-born males. So the sex chromosome composition for Turner syndrome, in which the individual has only one sex chromosome, is an X chromosome, and is designated class as X0 or XO or X0. So because they lack the male determining effect of the Y chromosome, individuals with Turner syndrome develop as females. However, both their internal and external genitalia structures are well underdeveloped, and they are sterile. So apparently that second X chromosome is necessary for normal development of ovaries in a female embryo. So examination of cells of these individuals reveals no bar bodies because there is no extra X chromosome to be inactivated. So using the standards of a bar body test, such an individual will be classified erroneously as a male. The incidence of this disorder class is 1 in 2,500 live-born females having Turner syndrome. So people with an X chromosome plus 2 Y chromosomes are phenotypically males, and they are fertile. Other characteristics of these individuals, tall, often with severe acne, hardly qualifies as syndrome. Hence the term XYY karyotype is used. Many years ago, several widely publicized studies suggested that males with this condition are more likely to display criminal tendencies and thus be in prison than those without this condition. However, these studies were flawed because they were based on a small number of XYY males without adequate or well-matched control studies of XY males. The prevailing opinion class in the medical genetics today is that there are many undiagnosed XYY males in the general population that do not have overly aggressive or criminal behaviors and are unlikely to be incarcerated. So there you have those. Next up, we get to abnormalities in chromosome structure cause certain disorders. So with this, we're getting to things such as the duplication, the inversion, deletion, and translocation. So chromosome abnormalities are not caused by changes in chromosome number, but also by distinct changes in the structure of one or more chromosomes. The breakage and rejoining of chromosome parts result in 4 structural changes within or between chromosomes. They are duplications, inversions, deletions, and translocations. So breaks in chromosomes are usually the result of errors in replication or recombination, and as is in a duplication, a segment of the chromosome is repeated 1 or more times, and these repeats often appear in tandem with one another. And you can see that class at A. This is figure 16.6a. And of course, these are A's. A. Duplication. The orientation of a chromosome segment is reversed in an inversion. For instance, as you see their class, instead of being a B, at that locus, it is now class BA. In a deletion, the breakage or loss of part of a chromosome along with the genes on that segment. So a deletion can occur at the end of a chromosome or on an internal part. So here at 16.6c, there is a deletion class of segment A. In some cases, a translocation of a chromosome fragment breaks off and attaches to a non-homologous chromosome. As you see here, instead of you having class A on this locus and B on that locus on that chromosome, it is now class B on that locus on that chromosome and A on this locus on that chromosome. So of course, in a reciprocal translocation, two non-homologous chromosomes exchange segments. So here we'll consider three simple examples of chromosomal structural changes in abnormal phenotypes: translocations, deletions, and fragile sites, which are chromosome sites that are susceptible to breakage. So translocation is that, of course, the attachment of one part of one chromosome to another. The consequences of translocations vary considerably. They include deletions in which some genes are missing and duplications in which extra copies of certain genes are present. In about 4% of individuals with Down syndrome, only 46 chromosomes are present, but one is abnormal. The large arm of chromosome 21 has been translocated to the large arm of another chromosome, usually chromosome 14. And then individuals with a translocation Down syndrome, of course, have one chromosome 14 and one combined 14-21 chromosome. So this class is, of course, what can occur: the reciprocal translocations. So a deletion is loss of part of a chromosome. So sometimes chromosomes break and fail to rejoin. Such breaks result in deletions of as little as a few base pairs or as much as an entire chromosome arm. As you might expect, large deletions are generally lethal, whereas small deletions may have no effect or may cause recognizable human disorders. So one deletion disorder is Cri du Chat Syndrome, in which part of the short arm of chromosome 5 is deleted. As in most deletions, the exact point of the breakage in chromosome 5 varies from one individual to another, and in some cases, Cri du Chat involves a small loss, whereas others involves a more substantial deletion of base pairs. Infants born with Cri du Chat syndrome typically have a small head with altered features described as a moon face, and a distinctive cry that sounds like a kitten meowing, hence the name, cry of the cat, in French. Affected individuals usually survive beyond childhood, but exhibit severe intellectual disability. The incidence class of Cri du Chat is 1 in 20,000 to an approximate class 1 in 50,000 live births. Let's go to fragile sites next. So with fragile sites, there are weak points at specific locations on chromatids. So this place at which a part of the chromatid appears to be attached to the rest of the chromosome by a thin thread of DNA. Fragile sites occur at corresponding locations on both chromatids of a chromosome. Both chromatids of a chromosome. So they have been identified on the X chromosome as well as certain autosomes. So the location of the fragile site is exactly the same in each one of an individual cells, as well as the cells of another family members. So in a fragile X chromosome known as Martin-Bell syndrome, the fragile site occurs near the tip of the X chromosome, and where the fragile X gene contains a nucleotide triplet CGG that repeats from 20 to more than 1,000 times. And a normal chromosome CGG repeats up to 50 times. And the fragile site can be seen in figure 16.7. Figure 16.7. And there you have it on page 249. Up next, we get to genomic imprinting. So with genomic imprinting, it may determine whether the inheritance is from the male or female parent. So some traits are the result of genomic imprinting or parental imprinting, which of course is the expression of a gene in a given tissue or developmental stage and is based on its parental origin. So that is based on whether or not the individual inherits a gene from the male or female parent. So with this, For some imprinted genes, the paternally inherited allele is always repressed, you all, or not expressed. And for other imprinted genes, the maternally inherited allele is always repressed. Thus, maternal and parental genomes have different imprints that result in different differential gene expression in the embryo. So now I'll get on into genetic diseases caused by single gene mutation. And you can review those two syndromes class based, or at least caused by genetic imprinting. Next, I'm getting into genetic diseases caused by single gene mutations. So what we'll get to here, class, would be those that are inherited as autosomal recessive, autosomal dominant, and X-linked recessive traits. So we'll get to phenylketonuria, PKU, sickle cell anemia, cystic fibrosis, Tay-Sachs disease, and then Huntington's disease, with, of course, hemophilia A. So you've seen several human disorders that involve chromosomal abnormalities. Hundreds of human disorders, however, involved enzyme defects caused by a mutation of a single gene. So these, of course, below will be discussed, and each of which is sometimes referred to as an inborn error of metabolism and a metabolic order caused by the mutation of a gene that codes for an enzyme needed in a biochemical pathway. And both PKU and albinism involve blocks in the metabolism of specific amino acids. So let us begin now. PKU. And as I get to this, just keep in mind that many genetic diseases are inherited as autosomal recessive traits. So these class we're getting to now are autosomal recessives. I say again, these we get to now are autosomal recessive diseases. So in other words, they're only expressed in a homozygous state. So why are these traits recessive? Well, most received mutations result in a mutant allele that encodes a product that no longer works. So in the heterozygous state, there is one functional copy of the gene and one mutated non-functional copy. The normal copy of the gene produces enough protein to meet the child's needs. In homozygous recessive individuals, both the alleles of the gene are non-functional and the cell's needs are not met. As a result, the person shows symptoms of the disease. Let's begin with PKU, phenylketonuria. So this class is a result of an enzyme deficiency. PKU, which is most common in individuals of Western European descent, is an autosomal recessive disease caused by a defect of amino acid metabolism. It affects 1 in 10,000 to 1 in 15,000 live births in North America. That's its incidence. So homozygous recessive individuals lack an enzyme that converts the amino acid phenylalanine to another amino acid, tyrosine. These individuals accumulate high levels of phenylalanine and phenylpyruvic acid and other compounds. So the accumulating phenylalanine is converted to phenylketones, which damage the CNS, the central nervous system, including the brain in children. The ultimate result in untreated cases is severe intellectual disability. An infant with PKU is usually healthy at birth, because a mother who is heterozygous breaks down excess phenylalanine for both herself and her fetus. However, during infancy and childhood, the accumulation of toxic products eventually causes irreversible damage to the central nervous system. In the 1950s, infants with PKU have been identified early and placed on a low phenylalanine diet, dramatically alleviating their symptoms. The diet is difficult to adhere to because it contains no meat, fish, dairy products, breads, or nuts. So although individuals with PKU should not consume the sugar substitute aspartame found in many diet drinks and foods because it contains phenylalanine. So biochemical tests for PKU have been developed, and screening of newborns through a simple blood test is required in the United States. It happens at every childbirth. They prick the child's heel and they determine whether or not he or she has PKU. Because of this screening program and the availability of effective treatment, thousands of PKU diagnosed children have not developed the severe intellectual disability. Most must continue the diet through at least adolescence, and doctors now recommend that the patients stay on the diet throughout their life because some adults who have discontinued the low phenylalanine diet experience certain mental problems, such as difficulty in concentration and short-term memory loss. Ironically, the success of PKU treatment in childhood presents new challenges today. If a homozygous female who has discontinued the special diet and becomes pregnant, the high phenylalanine levels in her blood can damage the brain of the fetus she is carrying, even though that fetus is heterozygous. Therefore, she must resume the diet, preferably before becoming pregnant. And this procedure is usually successful in preventing the effects of maternal PKU, which is especially important in women with PKU. Sickle cell anemia. So this results from a hemoglobin defect. So sickle cell anemia is inherited as an autosomal recessive trait, and the disease is most common in people of African descent. The incidence class is 1 in 500 African Americans. So about 1 in 12 African Americans is heterozygous. That is the incidence for heterozygotes. So under low oxygen conditions, a red blood cell of an individual with sickle cell anemia are shaped like sickles or half moons, whereas the normal red blood cells are biconcave discs. Half moon, biconcave disc. The mutation causes the sickle cell anemia was first identified in 1957. The sickle cells contain abnormal hemoglobin molecules, which have the amino acid valine instead of the amino acid glutamic acid at position 6, the 6th amino acid from the terminal end. So in the beta hemoglobin chain, that's where it occurs, the beta chain of hemoglobin. So the substitution of valine for glutamic acid makes the hemoglobin molecules stick to one another to form fiber-like structures, and that changes the shape of some of the red blood cells. This sickling occurs in the veins after the oxygen has been released from the hemoglobin. The blood cells is abnormal sickle-shaped, slows blood flow, and blocks small blood vessels, resulting in tissue damage from a lack of oxygen, essential nutrients, and in episodes of pain. So because sickled red blood cells also have a shorter lifespan than normal red blood cells, many affected individuals have severe anemia. So treatments for sickle cell anemia include pain relief measures and transfusions, and more recently medicines such as hydroxyurea, which activates the gene for the production of normal fetal hemoglobin. And this gene is not expressed after birth. Hence, of course, the hydroxyurea. The presence of normal fetal hemoglobin in the red blood cells dilute the sickle cell hemoglobin, thereby minimizing the painful episodes and reducing the need for blood transfusions. The long-term effects of hydroxyurea are not known at the time, but there are concerns that it may induce tumor formation. So ongoing research toward providing gene therapy for sickle cell anemia using a mouse model for the disease, and the first gene therapy treatments in mice used a mouse retrovirus as a vector, a carrier that transfers genetic information. However, the retrovirus did not effectively transport the normal gene for hemoglobin into the bone marrow, which of course is where hematopoiesis occurs, i.e. the stem cells producing red blood cells. The reason that sickle cell allele occurs at a higher frequency in parts of Africa and Asia and other parts of the world is well established. Individuals who are heterozygous carry alleles for both normal hemoglobin and the sickle cell hemoglobin, and they are more resistant than others to the malarial parasite, Plasmodium falciparum, which causes severe and often fatal form of malaria. The malarial parasite, which spends part of its life cycle inside red blood cells, does not thrive in sickle cell hemoglobin. when it's present. So an individual who is heterozygous for sickle cell anemia produces both normal and sickle cell hemoglobin. So in areas of Africa where the falciparum malaria occurs, correlate well with areas in which the frequency of sickle cell allele has been more common in the human population. Thus, those heterozygous individuals possessing one mutant sickle cell allele have a selective advantage over both types of homozygotes who may die of malaria or may die of sickle cell anemia. So this is a heterozygote advantage, which you all will hear about later on. So there you have it. Those sickle-shaped red blood cells clogging vessels. Now to CFTR for cystic fibrosis. So I've gotten to this before, but I'll say it yet again. Cystic fibrosis results from defective ion transport, and it is the most common autosomal recessive disorder in children of European descent, with an incidence of 1 in 2,500 births. About 1 in 25 individuals in the United States is heterozygous, meaning a carrier for that mutant cystic fibrosis allele. Abnormal secretions characterize the disorder, and the most severe effect is on the respiratory system, where abnormally viscous mucus clogs airways. The cilia that line the bronchi cannot easily remove the mucus, and it becomes a growth medium for dangerous bacteria. These bacteria or their toxins attack the surrounding tissues, leading to recurring pneumonia and other complications. Heavy mucus also occurs elsewhere in the body, causing digestive difficulties and other effects. So, as prior mentioned, the gene responsible for cystic fibrosis codes for CFTR, the protein that regulates the transport of chloride ions across membranes. So the defective protein found in the plasma membranes of the epithelial cells lining the passageways of the lungs, intestines, pancreas, liver, sweat glands, and reproductive organs. It results in the production of an unusually thick mucus that eventually leads to tissue damage. So although many forms of cystic fibrosis exist that vary somewhat in the severity of symptoms, the disease is almost always serious. Antibiotics are used to control the bacterial infections, and daily physical therapy is required to clear mucus from the respiratory system. So treatment with Dornase alpha, or DNase, an enzyme produced by recombinant DNA, helps to break down the mucus. And without treatment, class, death would occur in infancy. With treatment, the average life expectancy for individuals with cystic fibrosis is currently about 38 years. And because of the serious limitations of available treatments, gene therapy for cystic fibrosis is under development. So the severe mutant allele for cystic fibrosis predominates in Northern Europe, and another somewhat less serious mutant allele is prevalent in Southern Europe. Presumably, these mutant alleles are independent mutations that have been maintained by natural selection. So some experimental evidence supports the hypothesis that heterozygous individuals are less likely than other individuals to die from infectious diseases that cause severe diarrhea, such as cholera. Another possible example of the heterozygote advantage. Up next is Tay-Sachs disease. So this class is our last autosomal recessive disorder. And with this, Tay-Sachs disease results from abnormal lipid metabolism in the brain. So it's this autosomal recessive disease that affects the central nervous system and results in blindness and severe intellectual disability. Symptoms begin within the first year of life and result in death before age 5. Because of the absence of an enzyme, a normal membrane lipid in brain cells fails to break down properly. It accumulates in the intracellular organelles called lysosomes. The lysosomes swell and cause nerve damage, or at least cause the nerve cells to malfunction, Although research is ongoing, there is no effective treatment for Tay-Sachs disease. So the abnormal allele is especially common in the United States among Jewish individuals whose ancestors came from Eastern and Central Europe. So, in contrast, Jewish Jewish individuals whose ancestors came from the Mediterranean region, they have a very low frequency of the allele. Keep this in mind, class. Next class, we're changing gears just a bit because some genetic diseases are inherited as autosomal dominant traits. So with that, we'll begin with Huntington's disease, named after George Huntington, who was a United States physician who first described this in 1872. So it is caused by a rare autosomal dominant allele that affects the central nervous system. The disease causes severe mental and physical deterioration, uncontrollable muscle spasms, and personality changes. Death ultimately results. There is no effective treatment. Every child of an infected individual has a 50% chance of being affected, and if affected, of passing it on, the now abnormal allele to his or her offspring. Ordinarily, we would expect a dominant allele with such devastating effects to occur only as a new mutation and not to be transmitted to further generations. But because Huntington's disease symptoms typically do not appear until relatively late in life, with an average age onset of 40 years, affected individuals are likely to produce children before the disease develops. The incidence class here in North America is 1 in 20,000 live births. The gene responsible for Huntington's disease is located on the short end of chromosome 4, where it encodes a large 3,100 plus amino acid protein known as huntingtin. So with that, the gene, of course, appears later in life to regulate the formation of new neurons in the brain and plays a role in nerve cell connections. So with this, cloning the allele, became the basis for tests that allowed those at risk to learn pre-symptomatically if they carry the allele. And they, of course, have the decision whether to be tested for any genetic disease, understanding to be a high personal one. However, someone who tests positive for HD allele must then live a life virtually with the virtual certainty of eventually developing this devastating, incurable disease. So next we go, class, which is, of course, hemophilia, A and B. So with this now, this is an X-linked recessive. So we've done, now class, we've done nondisjunction being the cause with the Down syndrome, Klinefelter, and Turner syndrome. We've got two autosomal recessives with Phenylketonuria, Sickle cell anemia, cystic fibrosis, and Tay-Sachs disease. We did an autosomal dominant with Huntington's disease, and now we're with an X-linked inherited recessive, hemophilia. It was once referred to as a disease of royalty because of its high incidence among male descendants of England's Queen Victoria, but it is also found in many non-royal pedigrees. There are two primary forms of hemophilia, and both are caused by the absence of a blood clotting protein encoding by genes on the X chromosome. Hemophilia A is most common form involving clotting factor 8. Hemophilia B associated with clotting factor 9 deficiency. Both forms are characterized by severe internal bleeding in the head, joints, and other areas from even a slight wound. The mode of inheritance is X-linked recessive. Thus, affected individuals are almost exclusively male, having inherited the abnormal X allele on the X chromosome from their heterozygous carrier mothers. For a female to be affected by the X-linked trait, she would have to inherit the defective allele from both parents, whereas an infected male need only inherit one defective allele from his mother. Treatments for both types of hemophilia consists of blood transfusions and the administration of the appropriate clotting factor, the missing gene product, by injection. Unfortunately, these treatments are costly, and during the 1980s, many clotting factor preparations made from human plasma were unknowingly contaminated with HIV, and many men with hemophilia subsequently died from AIDS. Since 1992, virus-free clotting factors have been made available from both human plasma and recombinant DNA technology. Thanks a lot, RDNA. So, with this being the case, let's get on into the very end of things, and I guess I'll be here briefly, class, with X-linked genes and intelligence. So, geneticists are beginning to unravel X-linked genes affecting intelligence, and the X chromosome contains a disproportionate number of more than 200 genes identified so far to affect cognitive abilities. For example, coding for proteins required for the brain to function normally. So it's not surprisingly that many kinds of mental impairment are linked to defects in genes in the X chromosome. And by one count, the X chromosome contains less than 4% of the human genome, yet 10% of the genes in which defects are known to cause some form of intellectual disability are found in the X chromosome. And because males only have one X chromosome, more boys and girls have some form of mental impairment, a fact that has been observed for more than a century. So the gene therapy, here I'll be briefly. So with gene therapy, excuse me, because serious genetic diseases are difficult to treat, scientists have dreamed of developing actual cures using gene therapy. Those methods that are aimed to add or replace a normal therapeutic allele and its expressed protein for the defective mutant allele in the patient's cells. The rationale is that although a specific allele may be present in all cells, it may only need to be expressed in some cells or tissues to be an effective and potentially permanent treatment. One approach is to use a viral vector capable of moving into the normal allele into target cells isolated from the patient, such as the bone marrow cells, and that continually produce different types of blood cells. After verifying that the transformed cells express the normal gene, they are reintroduced into the patient to alleviate symptoms of the disease. So from here, we'll move on from gene therapy to the safety of such. And severe combined immunodeficiency. So the original gene therapy trials on humans were done in attempts to restore the immune system to children with severe combined immunodeficiency using the adenovirus vector. So some of the early trials of bone marrow were successful, but were temporarily shut down when a young man died in 2003 from an allergic reaction to the virus, and 5 children developed leukemia caused by the activation of an uncle gene. So it's yes, the safety of such. Potential toxicity of viral vectors too. All right, so from here, let's move on to genetic testing and counseling to end the chapter. Geneticists have made many advances in detecting genetic disorders in individuals in recent years, including in prenatal diagnosis and genetic screening. With these advances comes increased information for couples at risk of having children with genetic diseases. Helping them understand and deal with the genetic information now is part of the rapidly expanding field of genetic counseling. Prenatal diagnosis detects chromosome abnormalities and gene defects. So healthcare professionals are increasingly successful at diagnosing genetic diseases prenatally. In the early stages of pregnancy, of course, tests of maternal blood for fetal hormones and proteins that cross the placenta can be used to monitor for certain types of fetal genetic disorders. After 10 weeks of pregnancy, yes, it's at 10 weeks, testing of free fetal DNA in maternal blood is a screen for Down syndrome, trisomy 18, and trisomy 13. These tests have become highly accurate and are thought to detect 99% of all Down syndrome pregnancies. In addition, parents can be tested to determine if they are carriers for the same types of genetic diseases. By way of amniocentesis, a physician obtains a sample of the amniotic fluid surrounding the fetus by inserting a needle through the pregnant woman's abdomen into the uterus, and then to the amniotic sac surrounding the fetus. Some of the amniotic fluid is withdrawn from the amniotic cavity into the syringe. The fetus is normally safe from needle injuries because ultrasound imaging helps to determine the positions of the fetus, placenta, and the needle. However, there is a 0.5% chance, or at least a 1 in 200 chance, that amniocentesis will induce a miscarriage. So the amniotic fluid contains living cells sloughed off the body of the fetus and hence genetically identical to the cells of the fetus. After the cells grow in culture in the lab, the technicians karyotype the dividing cells to detect chromosomal abnormalities. Other DNA tests have also been developed to identify most chromosomal abnormalities. So this shows class how the amniocentesis is done. Researchers have developed prenatal tests to detect many genetic disorders with simple inheritance pattern, but these disorders are rare enough that physicians usually order the tests performed only if they suspect a problem. Enzyme deficiencies can often be detected by incubating cells recovered from amniotic fluid with the appropriate substrate measuring the product. This technique has been useful in prenatal diagnosis in disorders such as Tay-Sachs disease. The test for several diseases, including sickle cell anemia, Huntington's disease, and cystic fibrosis, involve directly testing the individual's DNA for the mutant allele. So amniocentesis is also used in detecting a condition known as spina bifida, in which the spinal cord does not close properly during development. The relatively common malformation occurring in 1 in 3,000 births, this birth defect is associated with abnormally high levels of the normally occurring protein, a fetal protein, in the amniotic fluid. So some of this crosses the placenta, and of course, could be why. Up next class is chronic villus sampling, because a problem with amniocentesis is that most of the conditions it detects are preventable and incurable, and the results are generally not obtained until well into the second trimester, when terminating the pregnancy is both psychologically and medically more difficult than earlier. Therefore, researchers have developed tests that yield results earlier in the pregnancy. Hence, I am here at chronic villus sampling. It involves removing and studying cells that will form fetal contribution to the placenta. Fetal contribution to the placenta. So CVS, which has been performed in the United States since about 1983, is associated with a slightly greater risk of infection or miscarriage than amniocentesis, but its advantage is that the results are obtained earlier in the pregnancy than amniocentesis, usually within the first trimester. Let's keep on getting earlier. But how this occurs, class, is you use a transabdominal sampling technique to withdraw the chronic villus cells using ultrasound probe, and of course, it can be done, class, by way of a cervical sampling technique as shown here. The syringe withdraws the chorionic villi cells, and of course, you culture those cells and test those by way of karyotype. So there you have it, catheter, class, and the chorionic villi. So next up, class, is preimplantation. Preimplantation genetic diagnosis is used to screen embryos produced by in vitro fertilization. So an embryo screening process known as preimplantation genetic diagnosis is available for potential parents who carry alleles for Tay-Sachs disease, hemophilia, sickle cell anemia, and dozens of other inherited genetic conditions. So PGD is an adjunct to assisted reproductive technology. The concept is by in vitro fertilization, in which the gametes are collected, eggs are fertilized in a dish in the laboratory, and the resulting embryo is then implanted in a uterus. So prior to implantation, however, the physician screens single cells of early embryos for one or more, I say for a number of genetic disorders, before placing a healthy embryo into the woman's uterus. PGD class differs from amniocentesis and chronic villus sampling, excuse me, in that the test is performed before a woman is pregnant, so it eliminates the decision of whether or not to terminate a pregnancy if an embryo has a genetic abnormality. However, PGD is not as accurate as amniocentesis or CVS and is more expensive. I would disagree with your text, class, and say it's even more accurate than both. Quite expensive, but more accurate, I would say. Moreover, PGD is sometimes controversial because potential parents may choose the gender of the offspring and not screen, and of course, not to screen for genetic diseases. So it could be used, class, by those who have a lot of money to choose a male or female child. Although using amniocentesis, CVS, and the PGD can help physicians diagnose certain genetic disorders with a high degree of accuracy. The tests are not foolproof, and many disorders cannot be diagnosed at all. Therefore, a lack of abnormal findings is not a guarantee of normal baby. Next in class, we get to genetic screening. So this is just the way in which you have a systematic search through a population, and with such, individuals with genotype or karyotype that may cause serious genetic disease, such as the PKU heel prick, or even sickle cell anemia, detects certain genetic diseases. And in adults, it's used to identify carriers of recessive disorders and help make informed reproductive decisions. So it's just if both prospective parents are heterozygotes, the carriers are counseled about the involved risks in having children. And since the 1970s, about 1 million Jewish adults in the United States, Israel, and other countries have been screened voluntarily for Tay-Sachs disease, and about 1 in 30 have been identified as a carrier. So Tay-Sachs screening programs have reduced the incidence of Tay-Sachs disease to almost zero. To genetic counseling. So couples concerned about the risk of abnormality in their children may seek counseling for medical or genetic information, as well as supporting guidance. So with that, the genetic counselor uses family histories to calculate the probability that offspring will inherit a particular condition. So the clinical geneticist screens for the detection of heterozygous carriers or certain conditions. So genetic clinics available in most major metropolitan areas are usually affiliated with medical schools, and the counselor, of course, receives training in counseling, medicine, and human genetics to provide people with the information they need to make those reproductive decisions. So they offer advice tempered with respect and sensitivity in terms of the risk estimates, that is, the probability that any offspring, or that any given offspring, will inherit a specific condition. So with that, we get to human genetics, society, and ethics. So recessive mutant alleles are present in all individuals and all ethnic groups, and each of us is heterozygous class for 3 to 15 harmful recessive alleles. Yes, us, you, me, us. So it's very unlikely that the abnormal alleles that one individual carries are also carried by their mate, with the exception of mating between close relatives, which I think you all heard of once before. I said heard of once before. So genetic testing also raises class a number of issues, meaning genetic discrimination even, or even just those ethics as far as should it be done. And of course it gets to those social issues and even legal issues. So should that genetic information be available to health insurance or life insurance companies or to employers? And physicians also then argue that people are at risk of particular genetic disease that might delay the testing because they fear genetic discrimination from insurers and employers. So since which class, the ethical, legal, and social applications research program of the National Human Genome Research Institute has developed principles designed to protect individuals against genetic discrimination. So it's called GINA, and it prohibits employers and insurance companies from discriminating based on information derived from genetic tests. And what I'll say, class, is ethically, at what age should genetic testing be permitted for adult onset diseases? Or what are those emotional and psychological effects that individuals who are told they have tested positive for an incurable disease? And then, of course, what should testing be performed when a family member wants testing and, of course, others do not? Should parents be able to test their minor children? Should access to the genetic test be permitted in cases where paternity or kinship testing? Or should states be able to collect that genetic data on their residents? And should school administrators or law enforcement agencies have access to the data? So with all of these questions, I won't give an answer. I'll just say that as we continue class into the future, these are things that I say we should all be quite aware of. And not just aware of, but know a lot about, because with technology class also comes a lot of questions because of things that can be done with science. This has been your instructor class, Skylar Huff. I hope you learned a lot. If you have any questions, feel free to contact me via phone, email, or even, of course, here in the office, or even before or after class. Thank you all for listening, class.